whole genome sequencing, and more to come in the future, I think. Yes.
Well, as a structural biologist, I should bring in at this point that we still interpret DNA only in terms of the linear sequence. We know that DNA is extremely densely packed in the cell, and nobody talks about this in everyday usage of genomic information. See, in proteins, we are long past the state where we would just talk about the sequence, but this is the way we talk about the DNA. And then only a few percent of the total amount of DNA in our body is understood in terms of cloning, encoding proteins or RNAs. Yeah. The majority of the sequence is
junk.
Junk DNA, they say.
Yes,
but and then some other people say it's not junk. You know,
there is no reason to believe that the majority of our DNA should be useless. And I think we have a lot to learn, especially about those parts of the DNA that we do not understand, and which is most certainly not just junk. Mm-hmm. And I think we have to understand in much more detail how the three-dimensional arrangement of the DNA and the chromatin impacts on the way the whole organism functions.
Maybe.
Is that...
well, I'm not so sure. I actually hate these people who say, you know, it's all in this, where the DNA is in the nucleus depend, you know, this is all important. It doesn't seem to have great explanatory power to me. You may be right, of course. I mean, and in some
it's too complicated
you know, and it's
to
sort
be understood. It's much easier to just read off a sequence of letters.
Yes, absolutely, absolutely. And it is basically a linear molecule. I mean, right? I mean,
proteins are also linear molecules.
Yeah, but proteins are a lot floppier, Kurt. You know that. I mean, they sort of, you know, they may be a piece of string, but they very quickly wrap up into a ball.
But the DNA is stiffer only because you always see the double helix.
Well, you can bend it a little bit, but I forget how much can it bend. People have measured how much it can bend. I remember.
DNA will bend about every 240 base pairs or so.
It's not all that. It is pretty stiff, actually.
As a double helix.
But you can bend it a lot more.
Single strands,
if you have single strand, then all bets are off.
So what you're saying is that the easy phase of linear DNA sequencing has been done, and now comes the hard phase of understanding how it's put together and used, and how the complex interactions there operate. I guess we understand a bit about how the proteins as transcription factors act on the DNA, but there's so much more to learn about how it all functions in the cell.